Welcome back to the Cambridge Neuroscience Interdisciplinary Seminar Series. This series features current work from neuroscientists across the schools and departments of the University of Cambridge, reflecting the pioneering work and diverse interests of our members here. Each seminar focuses on one of the new interdisciplinary themes of Cambridge Neuroscience, and for more info on the talks covered in this seminar series and all things neuro-related here at Cambridge, please follow us on Twitter at CamNeuro and follow the links below. Good afternoon, everybody. Welcome to the Cambridge Neuroscience Interdisciplinary Seminar Series. Today, we are delighted to welcome Professor Michelle Ellefson. Michelle is a professor of cognitive psychology at the Faculty of Education, where she convenes the Instruct Lab. And I actually had to look up this acronym, but it is the Implementing New Student Thinking Resources Using Cognitive Theory. And her whole idea, and I think it's really epitomized in her graphic here on her opening slide, but her work really combines cognitive psychology and educational neuroscience. So we're delighted to welcome Michelle today because it's something a little bit different that I feel quite strongly about, open science and credibility in neuroscience and all the efforts being done by many organizations, including, say, for instance, the British Neuroscience Association. So we're really happy that Michelle is with us today. Over to you, Michelle.
Great. Thank you so much, Derb. Thanks so much for the invitation, and to all of you who are here today at the talk. I think it's really great that we can do still a handful of online talks, because I think it opens up, the kinds of work we're doing. Derb, you said all the things I was going to say at the beginning, so I'm going to say instead I'm going to pick a little bit on the name of the lab, because that is an acronym that the person who I'm collaborating with this work that I'm presenting today actually came up with, and then we worked out the specific letters that fit in. So my background, I was trained in brain and cognitive sciences, but as I was going through my training, I became really interested in this intersection between what we know about thinking and reasoning and learning from a cognitive psychology, cognitive neuroscience point of view. How does that link to what's happening in school settings and decision making that happens in schools? So my work is really very much at this intersection, and I kind of wanted to have a lab group that reflected that idea, that was that bit. So today I'm going to talk about some work that I started really during the pandemic with a colleague, Danny Oppenheimer, who's at Carnegie Mellon. You'll see his photo later when we get to the actual study itself. And this is work that we did linked to a series of conversations. And because it happened during the pandemic, it kind of went through the idea generation, the data generation, and writing about publication without actually sharing it anywhere in a conference, because we aren't really going to many conferences. So this is, even though this is all work that's fully published, it's the first time that I've talked about it. So I'm looking forward to seeing what people think and what kinds of questions you've got about it. And we had a handful of questions from reviewers, but it went through so easily that I'm still not sure I understand where the critiques may be. So I'll be waiting for that at the end. So I'm not entirely certain about all of the audience, but I thought it was good to just do an overview of kind of how I see this general idea of open science. And I tend to think of good science as something that is robust, reproducible, replicable, and generalizable. And I love this graphic. It was created for the Turing Way, and it's openly available for use, because I think it helps to think about those different contexts. And I'm definitely taking this sort of approach in terms of the vocabulary that I'm using. So reproducible data is where you have the same data and the same analyses. And we would hope that in our work that happens. I mean, we do that ourselves in terms of we've run the data and then we're rechecking it as we're getting ready to publish it or share it. We want to make sure we actually have the same, we haven't made any mistakes anywhere. So that's something we probably hopefully do even ourselves, although there are conversations about how reproducible published work actually is in terms of somebody else replicating the analysis. Robust is where we have the same data but different analyses. And again, this is something we've really done for quite a long time as researchers, which is thinking about, if I try something a different way, especially when we have a lot of complex models, making sure that we get a similar sort of finding. So findings that show up repeatedly, even though we might be analyzing things in slightly different ways because there might be slightly different analyses that are appropriate. That's also something a lot of us look for before we publish, and we often ask each other questions in the robust arena when we go to talks, and I imagine there may be some of those questions today when I present some of our data. The other side is an area where I think we have, well, the bottom right-hand corner is something that's also been going on for a long time, which is we think about generalization, which is, so I've got this finding and then somebody else tries to extend it into a different sort of setting using different kinds of analyses. And there's a lot of that, I think, that was happening before the so-called kind of concerns about replication was, we were trying to generalize things. And sometimes things would generalize and sometimes not. And we didn't always know if that was because the generalization wasn't possible or because there was a problem with the research itself. And I think the upper right corner is, at least in areas that are linked to psychology, so cognitive psychology, cognitive neuroscience, is where we've seen a lot of advancements in the last decade, which is thinking about replication. And that's where we would have different data, but the same analysis. And this is where we try to actually replicate what someone else has done. And this is the space that this particular work comes from, because it's thinking about what is actually a good replication. And sometimes that's easy because researchers put all of their materials online, or they're using some kind of experimental program that they can just share, and you can do the exact, or you think you're doing the exact same study. And sometimes it's harder because there are lots of things that might happen in the course of any bit of data collection that may be hard to replicate fully because there are changes that occur. And so it's in that upper space that I'm going to get us to think a little bit, but I'm going to back up and do some of the overview around, why are people concerned about replication, and what are the things that might be problems. So in the conversation around open science, there have been a variety of different studies. I have to go the correct direction. My mouse is, there we go. A few different studies that have talked about this idea. So this is a paper by Baker in 2016 in Science, and this is asking scientists from a variety of fields whether or not they've been able to reproduce an experiment. So this is in that replication space. And if you look at those bars, I mean, they're not great, right? So we would hope that we can reproduce our own work at 100%, but that's not, even our own work across different disciplines, it doesn't look like that's the case. And in that same study, they asked scientists to report what they thought was contributing to research not actually being replicated. And these are the sort of things for people who have been in this conversation already that we're concerned about, things like pressures to publish, selective reporting, not having enough oversight or mentoring, methods or code are unavailable. So this is where we've had the move to putting our data and our code online. And then, of course, there are other things like fraud or issues with peer review. But these are kinds of conversations, I'd say, last decade or a little bit more, that scientists have been having more broadly, not just in psychology or neuroscience. And now, way back to 2012 in psychological science, John et al. Asked a bunch of psychologists how often they engage in some of these practices that we think might be concerning. And this particular bit of data shows the dark bar was the self-admission rate from people that they surveyed. And then they had a few estimates that they modeled based on some information and their willingness to trust that people would actually admit to what they've done. And these are the same sorts of things that come up in the previous slide. Things like, not reporting all of the dependent measures, not reporting all of the conditions. So that's kind of a selective reporting sort of thing, rounding down p-values, or, selectively reporting the studies that work, and the studies that don't work, they end up in a file drawer, they don't get published, they just go somewhere else. And this sort of conversation has led to some concerns about the published literature being biased, and it's not actually representative of what's actually going on in terms of findings. So a recent study in Royal Society Open Science by Hardwick et al. Looked at, well, how reproducible are the findings that we have. And this is just looking at reproducing the same analyses with the same data. So the data are available, the code is available, or maybe not, but there's enough description. And they had a number of studies that they did. This was in, I think, psychological science, if I can remember correctly off the top of my head. And I think what's kind of good news about this, so I think there's a concern in terms of things not necessarily being the same across different attempts at a bit of research study or different analyses, but there on the diagonal here is what you'd expect if the research is replicable based on what was found. On the x-axis, they have the original p-value. So this is just looking at the p-value itself. And then on the y-axis, they have a reanalysis p-value. So if a study is fully reproducible in terms of same data, same analyses, but somebody else can do it, then it should fall along this diagonal line. And actually, most of the studies that they reviewed did that. And there are a few that didn't. So there are some that had some minor discrepancies, which are the circles, very few of those, just a handful. Then there are a few more that had some major discrepancies, and there are a couple where there was some sort of decision error. And this particular study goes into a bit of detail around some of the reasons why they might have had those discrepancies, and also some of the challenges of getting the actual data and making sure that the initial analysis was actually reproduced appropriately, because sometimes not all of the information is there. But when they did get the information, I think it's kind of encouraging how much is showing up as being reproducible. Same data, same analyses. My mouse is going the wrong way. So this is a really new study, which I found really interesting. So this is starting to think specifically around psychology and how replicable the different branches of psychology might be. So this is a study by UU. It's actually just published in the Proceedings of the National Academy of Science. And what they did, looking at thousands of published papers across the literature, they used machine learning to estimate how likely was a given study to be replicated. So this is now in the idea of different data, different analysis, different data, same analyses. So you're actually trying to run the study again. How likely was it to be replicated? And this particular graph compares that against the experimental nature of the field. So for example, I'll pick on my own field first. And so this is cognitive psychology. So those of you who know cognitive psychology or cognitive neuroscience know that it's quite an experimental field. Most of the studies involve an experimental manipulation of one or another. So it ends up being on the right-hand side of the x-axis because there's quite a lot of experimental work. Where another area of psychology, like let's say clinical psychology or even personality psychology, are much more observational studies. There's a lot more survey research, correlational analyses, that sort of thing. So they end up being on the left side in terms of experimental research. And this particular graphic, what they were looking at is the kind of the relationship between the predictability in their machine learning simulation and the experimental nature of the field, to kind of get a sense of that overview. So one of the things that their results were kind of suggesting is that experimental work is actually kind of harder to replicate than non-experimental work. And there may be some really important reasons for that. They haven't gone into a lot of those reasons, and I'm hoping maybe I'll get to highlight some of those a little bit with Fidelity. And the exception is probably developmental psychology, but one of the issues for developmental psychology is that individuals are studied across time, across different ages, and there may be just some complexities for developmental psychology that make it kind of harder to fit on that nice sort of summary of experimental work actually being harder to replicate than non-experimental work. And I think this is probably, I mean, this is a new paper and there may be lots of different reasons why they might have found this. Remember, it's a simulation based on lots of existing papers. So this isn't necessarily where the field is, but it's an assumption based on the kinds of information that's available in the papers and the kinds of data that they're using, how replicable they might be. So a few years ago, Minofu et al. Did a really nice kind of manifesto for open science, and they identified these different kinds of threats that I've been summarizing in lots of different kinds of studies, but they put them in one place, the threats to robust, replicable, reproducible, and generalizable research. To be fair, that's not the word they used, but I think it follows those 4 parts of the graphic I showed you at the beginning. And so all along the kind of research pipeline, there are potential problems. So some of those are not controlling for various kinds of bias. They may be things around the design itself, the way the research is run, the way the analyses are run, and also just what's actually existing in the literature. So I'm going to give a little bit of time to each of these so that we can kind of see how people are conceptualizing them. And I've chosen simulations because the data I'm going to show are also simulations to try to kind of show, how people are thinking about these questions and how they might matter on a big scale. So p-hacking is the first one I'm going to talk about. Anybody who's been thinking about open science has probably heard a bit about p-hacking. P-hacking is this notion of, manipulating something with the analysis or how you interpret the analysis to get the p-value under, the happy threshold for publication. And I say that kind of maybe in air quotes. But there is this general notion, if something is statistically significant, if the p-value is less than 0.05, then it's publishable. And if it's not statistically significant, then it isn't publishable. That's kind of an old notion. So that may, at some level, drive researchers to make decisions about their analyses and what they publish. So this is kind of where cherry picking comes in, to basically fit under that umbrella of, well, this is statistically significant. And Uri Simonson has done a lot of work talking about p-hacking. And this is from a recent blog that Uri did, which is, I think, a nice little graphic showing that there are different kinds of p-hacking. So some p-hacking may have the impact on the p-value is actually small. So the changes in p are smaller, and he calls that slow p-hacking. And that may be things like, well, eliminating a few observations, tweaking the definition of variables. This may be, this is potentially things about how many participants, what do we do with missing data that can have an effect. Then there's also something called fast p-hacking, where the effect on p is actually bigger. So it's things like testing subgroups, cherry picking arms or conditions. So, not reporting everything, testing interactions that you hadn't planned for, or using, additional kinds of analyses that you hadn't anticipated. And what Uri has said in this blog is actually, so fast p-hacking has a greater effect on p, but it doesn't show up as much potentially in the literature as the slow p-hacking. And that's what these histograms represent. So the x-axis is a z value. And the idea is that z's that are, let's just say for rounding error, greater than 2, it's not exactly 2, but let's just do rounding error. Z's that are greater than 2 are tend to be statistically significant. And so in a lot of studies, what people do to try to harmonize across the studies is they turn any kind of analysis into a z-score to be able to make sense of what is statistically significant across lots of different kinds of studies that may use lots of different kinds of analyses. And so what Uri is suggesting is that for slow p-hacking, this is where we're just tweaking little bits. So we might be going from something like p is 0.06 to p is 0.045. This is where we tend to see more bunching around that statistical significance end. And so this is kind of potentially, researchers going, oh, it's so close. What can I do to get it over the line, sort of thing. And so there's a bit more bunching with the slow p-hacking. Where the fast p-hacking, there's less bunching around statistical significance, but it still has an effect because it still means, like, stuff is left out. There's there's this, selection sort of bias in terms of what is actually getting reported. So other kinds of things that were in the graphic that I mentioned about the threats to robust, reliable, replicable, and generalizable research is a control for bias. Harking kind of falls into this. My graphic's not a perfect example of harking, but it's linked. So harking is basically deciding what your research questions are after you see the results. And it's changing your view after you see the results. So this is an example of, so in 2000, there became a requirement for work where people were doing clinical trials that they had to be registered before the trial occurred. And so what happened is, if you look back on this sample of publications from the 1970s until mid-2014, is that before this requirement to register trials was put in place, there was kind of a mixture of what was published that was null and benefit. And actually, there were many more clinical trials showing benefit being published than those that showed a null result. After clinical trials had to be registered, you see an increase in the number of publications of things where there's a null finding. And actually a smaller number of papers where there's a benefit. And so the idea, this is where the harking kind of comes in, which is basically you don't change the idea once you see the data, but you start with, well, this was the intervention, this was the clinical trial that we were running, and it doesn't matter what the result is, it's interesting regardless. And so this has been part of the change as well in terms of thinking about, well, what should be published. But it has meant that older literature might be biased because it's then only things that might show a benefit that get published rather than those where there's a null finding between some sort of trial and control group. So publication bias is linked in all of this as well. So I mentioned things about having bunching up around statistical significance. So this is the same kind of graphic of a Z statistic, and where we've got kind of this high propensity of p-values that are very near statistical significance. And this is kind of that statistical stuff gets published. But Kuber also has some other work, which I think is important because this is linked to another element on the graphic I showed you by Manofo et al., which is that one of the links in terms of in terms of the threats is the sample size. Do you have enough statistical power? So this has also been a big change in the last 10 years. We're not really seeing publication of small sample sizes, and even in neuroscience with things like imaging, there's much more almost collaborative work and trying to do bigger projects with bigger sample sizes across multiple labs, because of course imaging is really expensive, so small sample sizes have been okay, but seeing the problem of small sample sizes has led to a lot more collaboration. And this particular paper for 2014 and +1 shows how there's some controls that they did here, but how the sample size and the effect size actually have a negative correlation. And so this is suggesting that when we've got small sample size, we may have larger effect sizes, which may mean we're inflating the actual statistical significance or the effect size, and that makes it potentially harder to replicate later. Okay, so going back to Manofo's lovely graphics, so I've done a really kind of a whirlwind tour of the kinds of things that they were talking about as important to thinking about replication. And now I'm going to introduce something that's a bit new. And this is what came from conversations that Danny and I had. So it was actually in the first year of the pandemic, we were talking a bit about an increase in the use of replication as part of undergraduate research methods training. So an undergraduate research methods course, all of the students might take part of a replication of a study. They may choose a study and then they'll work on replicating it. And although that's really good from a pedagogical point of view, what might that mean in terms of the science? So this was the initial conversation between Danny and I about, well, if you've got a lot of naive researchers trying to replicate something, how faithful will they be to the actual kind of procedures that need to be done? And in that conversation, my experience with education, and those of you who might be in the audience who know about clinical medicine will know about the term fidelity, or sometimes treatment fidelity, which is this idea of a consistency in delivery across all all participants or all conditions, depending on what the conditions are, but there's a consistency. And we started to wonder if what we were seeing in terms of data on replications, and how the most oftentimes replications have smaller effect sizes than the original study. And we started to wonder if some of that reduction in replication could be linked to some of the conversations that happen around fidelity in other fields. So it was kind of an application of educational research, intervention research, medical research into kind of a cognitive psychology space. So this is a paper from 2015 from the Open Science Collaboration Group. And they were basically looking at the link between the original effect size and the replication effect size. And so this graph on the lower left is, I quite like this graph. So basically the y-axis is the replication effect size, and the x-axis is the original effect size. If it's green, it means it's statistically significant effect size. If it's red, it means it's not statistically significant. And then they plotted those against each other in terms of correlation. And they also use the size of the dot to indicate the statistical power. Now, there's, again, a positive correlation. So, the larger the initial effect size is, the more likely something is to replicate. But there's still a lot of papers that were statistically significant in the first iteration that aren't in the second iteration. And those effect sizes are going down. And we were thinking about, well, what does that mean? There was a paper and some work by colleagues who do educational research, Neil Humphrey is one of those, where they were finding this common phenomena when you're looking at educational interventions, that when the interventions are conducted by educational researchers, they get larger effect sizes than when the interventions are conducted by teachers. Now, there could be lots of interpretations of why that might be happening, but one thing I think that could be driving that is the researchers understand a large number of the details of why certain things are in place in terms of the intervention, where the teachers who may not have designed the intervention are not necessarily aware of all those really subtle things that could play a role. And so this is where we started to think about, is this something we might be able to model? Could we simulate some data to actually look at this across a wider scale, across a variety of kinds of levels of fidelity to see whether or not what impact is fidelity actually having on something like effect size? So this is a photo of Danny. This is our paper. It was an online first in psychological methods. So there's some information about the DOI as well as OSF. I have to admit, we didn't pre-register it because we had no idea what we'd find, but we decided once we had sorted out all of our scripts that had to be all up so people could change the values that we decided and see how robust these models might be. So fidelity is broadly conceptualized as a consistency across implementations. And of course, as I've said, a lot of the work in fidelity and thinking about fidelity happens in clinical science and intervention science. But what Danny and I started to think about is what might be the potential threats to fidelity in an experimental cognitive psychology sort of study. And we thought, well, there are probably two things that may matter in terms of fidelity threats. So one might be the research team. So I was kind of alluding to that when I was talking a little bit about the difference between researchers and teachers running educational interventions, and also like the conversation around, well, what happens if you have experienced researchers versus novice researchers running studies? So there's probably a number of things that might be linked to kind of the research team, how comprehensible are the methods? What about the demeanor of the person who's running the study? So if, in a classic cognitive psychology or even cognitive neuroscience sort of setup, you've got participants who come into a lab, and what could be the effect of somebody who's really welcoming and puts everyone at ease versus somebody who's not as welcoming. So the demeanor of a researcher might have an impact on how a participant, engages with the research, and the diligence of the research team. So some examples that we have from our own research that we pulled out. Danny talks about how one time he had an undergraduate research assistant who was running a study that they had, and who ran the study in the dark because the instructions did not say turn on the lights. And so they had to actually get rid of all of the data that was run in the dark because they knew that there was, a problem in terms of the administration. But that's, kind of a clear example of where there's been a deviation of the method. For me, I've reported this actually in another paper where we talk about applications of cognitive developmental psychology to education, where we had somebody who was helping us do data collection who was a former teacher. And although in our training, we were doing a standardized assessments where you had to stick very much to the script and you couldn't help participants get the answer. They get more and more complicated as you go along. And despite having a training that was very much regimented, we noticed that that this particular RA was coaching the children, trying to help them get the answer, which is very much a teacher frame of reference. There's no critique of that for teachers because teachers are very used to coaching children to do better, but that's not quite how we do things in a cognitive psychology or developmental psychology sort of place. So you can have these kind of different experiences of researchers. There's also a research environment can matter. So things like the timing, is research happening right after a big lunch, or is it really early in the day or late in the day? Temperature can be a big thing. So when I was a graduate student, we always had this time of year when the heating was still on in the spring, but the temperature was hot outside, and it would get so hot we'd just have to cancel any kind of data collection because it was 35 degrees inside because of this combination of it warming up outside. It's probably about 20 to 25 outside, but inside it's really hot. And so those sorts of things matter in terms of the kinds of things that might be happening from one bit of data collection to the other, and other kinds of distractions. So Danny had at a previous university, his lab was right next to the field where the marching band in the US practiced. And so they had to actually stop scheduling any of their research experiments during that time because it was so loud, everyone could hear it, and it was quite distracting. My favorite one is a study that I did where we're looking at chemistry reasoning in young children, and we're looking at how they reason about mixing. And just as kind of a prompt to see what they thought before we did the mixing, we would ask them what they thought might happen. So we showed them some water, and we'd show them something like, let's say, sugar, and we just say, well, what do you think will happen if we mix them together? And we had one school where we were in a little quiet side room, and on the wall next to us was a food pyramid. And we had a few kids who they were reception age, so they didn't really know what to say. And they looked at the picture and they'd say, it'll turn into an apple. And then the next time, it'll turn into a banana. But if we weren't, we didn't actually end up using that data because we had other things, we used other bits of the data. But because we were in that room, they were coming up with ideas that if we had just had a blank wall and we weren't in a schoolroom, they would have just said, maybe, I don't know. So the kinds of environment that you're working in can really matter, and you don't even know, what kind of environment a participant might be walking through to come to a study as well. So these are some examples that we thought of where fidelity can get mixed up in terms of researchers who might not necessarily link to the task, but also just things that might be happening in the space where the research is occurring. So that was our impetus for trying to model this. So we did 3 sets of models. I'm going to try to walk you through slowly the first model, and then we'll kind of get, we specifically did some iterations. So we wanted to try it in this first study to just think broadly, big samples, what kinds of things might be happening in the different kinds of scenarios that people might be working, and how can we vary the values of things like fidelity or change. So in model 1, we thought of this like having 40 classrooms. Model 1 is a little bit more of a classroom-based idea. So you have a classroom-based intervention where you might have a teacher who is following a protocol or not. So the fidelity is good versus bad. So we had 40 classrooms. We have 1,000 participants. And that means there are 25 students per classroom. So this is not necessarily perfect, but we thought this was a good kind of way of thinking about the sample size. And basically, this is a simple experiment versus control sort of setup. So the assignment to whether it was the experiment or the control group was 50%, the probability is 50%, and the assignment was by teacher, so by classroom. Then what we did is we made the assumption that we were going to generate data based on a mean score of what we called base ability, and that value was going to be 100. And this is kind of arbitrary, but I thought 100 was quite good because we can use that in other places in terms of thinking of proportion. And the standard deviation would be 10. So that's the value, that was how we were going to generate new data, was based on the assumption that a mean score was 100. I'll show you the different ways we generated data in a minute, and the standard deviation was 10. And then what we did was we then had various kinds of different models where we varied how much the experiment would change that ability score. And we thought of it as ability because we were thinking about it in a classroom sort of setting. And it could range from 0 to 20. And that's basically because that goes two standard deviations above and below. No, well, it's one standard deviation, no, two above and two below. So we kind of went from 0 to 20, and we sampled what seemed like a reasonable number in between those ranges. And then if the assignment was to the control group, then the ability change was 0. So we made an assumption that a control group saw no change, which is of course an assumption that's pretty clean, but we just decided, let's just make it simple. Control group, no change. Experimental group, you have a change from 0 to 20. And then we also had a different range in terms of potential ranges of fidelity. So fidelity, we thought of as a percentage. So what is the percentage that you're adhering to the protocol? And we couldn't really do the perfect like 100% or 0 because that creates some problems in terms of the mathematics. So we went from 0.01, which is kind of like 0% fidelity, up to 0.99, which is kind of 100% fidelity. And then we just sampled at intervals of 0.2 in between there. And then we also varied the standard deviation of fidelity. So what we basically did is we took all of the different combinations of these different values, and we reran different simulations of every combination of what change happens in ability, what's the fidelity value, and what's the standard deviation fidelity. We also had some constraints, like we didn't think, well, fidelity can't ever go over 1. So we didn't sample, for example, a fidelity mean of 0.99 and a fidelity standard deviation of 0.99. So we kind of thought about trying to keep things within the 0 to 1 range as well. So there were some combinations that weren't really viable. And I'll show you on the next slide, what basically happens is that we have a total of 165 different simulations that come from this same model. So it's like creating 165 different experiments where you have a different change in ability in the intervention and different average fidelities and standard deviations. And then we get an estimate of data around that that's generated. So it's like running 165 different experiments with 165,000 participants. So we're able to look at this without having to worry about issues on sample size. Now, the last thing that we didn't do in the first model was deal with this idea of classroom bias or lab bias. And this is where some of the things we were talking about in terms of you might have, within a classroom setting, you might just have some classrooms that are better. You might have teachers who are more experienced. You might have children who come from higher socioeconomic backgrounds. So we kind of added, which you'll see in the second and third model, this idea of just some sort of bias, the stuff you can't really control, but that might have been outside of, that might have been impacting things. So if we think of some of the examples of, a participant is coming into a research lab, we don't know what's happened that day that might distract them when they come in to participate in the experiment. So we kind of had this measure of some kind of bias. Okay, so that's the set of model 1. Now in models 2 and 3, we add in classroom bias. So the first kind of parameters, they're all the same, but we now sampled the potential classroom bias across a range following a similar logic to what we did in the previous model. And then model 3 has one difference, and that is our first two models were very much based on kind of a classroom approach, and model 3 is based on almost a many labs approach, which is happening in terms of research, where you're seeing lots of different laboratories are doing a replication. So you might have 10 laboratories or each collecting 100 participants, and then all that data gets put together. So we wanted to do a model that was a little bit more like we think of as a cognitive psychology sort of replication. And so this particular model is exactly the same in terms of the various parameters that we're testing. But the difference here is that being assigned to experiment versus control group happens at the participant level, and then the number of labs, if you will, is now 10. So you have 100 participants per lab. Okay, so that's the basic kind of all the various parameters that we were testing around. I mentioned earlier that in model 1, we end up with 165 simulations. In model 2 and 3, because there's a lot more that we're trying to do every viable combination, we have about 5,500. So this is representing about 5.5 million participants. So this is, anybody who does simulation work, the great thing about simulation work in this sort of space is that you can do it without having to worry about sample size, and you can really get a sense of, the broad range. I'm going to report data using a Gaussian distribution because we made an assumption that we would have a normal distribution of data. We tested this using a variety of other ways of generating data: uniform, Poisson, exponential, binomial, and random distribution, and we get pretty much the same result. We have the script for the extra different kinds of distributions available on OSF, but the main part of the manuscript just presents the Gaussian. As I mentioned, each simulation then includes 1,000 participants. And then what we did in terms of thinking about the outcome is we focused on this idea of effect size, because that's, of course, what a lot of people are thinking about. So for each participant, we calculate a final score, which is based on their base ability, which was randomly generated through the Gaussian distribution, whatever their lab or classroom bias is, which would have also been randomly generated based on the Gaussian distribution of the combination of classroom and lab bias, plus the product of fidelity times delta ability. Because if what we made the assumption is that if the intervention is supposed to change ability, let's say by 10, but you're not getting all of the elements of the intervention being delivered to you because the fidelity is not 100%, then you're going to get less of that benefit because the fidelity is not all there. And then we basically compared the effect sizes, which was basically a final scores of the intervention versus the control group. And we had a few other ways that we had kind of controlled for that in terms of how we looked at intervention versus control. And then computed basically Cohen's d between the two. So what ends up happening is for model 1, we have 165 bits of data that we're then plotting effect size versus the parameters that we've got. So that's how I'm going to show you the data. Fair warning, now we're going to see a huge amount of correlation matrices. So I just really like this output in R, and it's maybe a little bit much, so I'll draw your attention to the key bits. But okay, so along the diagonal is the histogram. So you can now see the values that show up. And for the data that we are generating, you can see the we're getting kind of this normal distribution with some caveats based on how we're sampling the space. Below the diagonal is the scatter plot for the variables. So this is fidelity mean versus fidelity standard deviation. So we can be transparent about how is the mean and standard deviation relating to each other. They shouldn't, they're going to be related a bit because of the way means and standard deviations are linked to each other. And then above the diagonal is just the correlation. So the key correlation is this one up here at the top in the middle, which is the correlation between fidelity and effect size. And so in our model 1, we get a correlation of 0.64. We subsequently ran some regressions after that to look at, what's the kind of if we think about predictability of what's the effect or the influence, let's say, of a change in fidelity and a change in effect size, and our regressions indicated a 1-to-1 effect. So for every 1% decrease in fidelity, there's a 1% decrease in effect size. But that's not uniform across the spread of the data. There's actually a bigger influence of fidelity at larger effect sizes than at smaller effect sizes. Okay, so that's just kind of that ordinary model. What happens if you've got things like, if we count for all these other things that participants might be going through, or classroom bias, if you will, and we get a similar finding. The correlation's a little bit weaker. So here's the correlation, it goes down to 0.54. But what's mostly interesting is to think about how something like classroom bias kind of interacts with the general notion of fidelity and effect size. And what we did is we did a series of partial correlations where we controlled for the mean of the intervention group on the link between classroom bias and effect size. And what we found was a negative correlation across all, between this model and then the next model, model 3, indicating that the heterogeneity in things like classroom or lab factors have less of an influence on effect sizes than does fidelity. So fidelity and how well researchers are adhering to the protocol has more of an influence than some of the extra factors that might be going on. Okay, so I'm going to show you the last slide and just kind of say, like, this isn't that different from the other two models. The difference with the model 3s is that we decrease the number of labs and increase the number of participants within the labs. And we're seeing,
You've gone the wrong direction.
I did go the wrong direction. I should have, my mouse does not like me. Sorry, everybody. This is it. So what happens in model 3, this is where we've just kind of thought of it a little bit more like the many lab situation, is that we're seeing a very similar story to what we've seen in model 2 with classroom bias and also with model 1. So our, similar sort of correlation between fidelity and effect size. Okay, so I'm going the correct direction now. Right, so the key lessons that we've learned from this first stab at thinking about fidelity and the kinds of things that might impact how well we are implementing research is that replications could fail even without things like p-hacking or other kind of intentional things that researchers are doing, down to things like fidelity. So lack of fidelity could actually reduce how well a finding is replicated. And the variations in fidelity are leading to a heterogeneity in findings in terms of this sort of research. And this is linked to some other work that people have been doing. So there's some really nice meta-analyses, like by Stanley et al. From 2018, or some of the many labs replications. An example might be a more recent one by Klein et al., which is basically this idea that the more variation you have in how research is delivered, the more chance you take on a replication of that finding not working. And I suppose one of the claims we've made in the paper is that we've done quite a lot of tight controls here. We used a limited number of input parameter types, and so it could be that our estimations of that 1-to-1 correspondence could actually be underestimating how much fidelity might influence findings in the real world. But we're okay with an underestimate. And it would be nice to see some other sorts of models into the future. And as I said, people can go to our script, download it and play. You can put in any sort of parameters you want and see what happens as well. So that is the end. And so a quick whirlwind tour of fidelity and just general kind of open science. And I suppose one of the things that we said in the paper that will be my kind of wrap up is that even though there's a lot of conversation in intervention science work or in educational work around the importance of fidelity, and that we should be reporting fidelity or we should be measuring fidelity, even in fields where fidelity is kind of a common word, nobody really measures it. So Danny and I did kind of sneak in the discussion. We think like it would be great if we could start to measure fidelity and think about it as a metric in experimental research, so that we can actually utilize it, maybe having a checklist of the things that we have to do to utilize it as we go forward in terms of thinking about how to keep our protocols consistent across various kinds of situations that we might have in data collection.
Thank you, Michelle. A really different type of seminar for us and one that is so important. Join us next time when we welcome Professor Valerie Voon. Valerie is a professor in the Department of Psychiatry here at the University of Cambridge and at Fudan University in Shanghai, China. She's a consultant neuropsychiatrist with the Cambridgeshire and Peterborough NHS Foundation Trust, and her group investigates mechanisms underlying self-control and repetitive behaviours and the influence of emotion and incentives. Her lab focuses on integrative neuromodulation, identifying modifiable biomarkers using multiple modalities, with the goal to optimise and develop novel means of neuromodulation. She'll be delivering a seminar entitled Integrative Neuromodulation from Biomarker Identification to Optimizing Neuromodulation. For more info on the talks covered in this seminar series and all things neuro-related here in Cambridge, please follow us on Twitter at CamNeuro and follow the links below. See you next time.